Hello my dear students, this is Amandeep along with Monica ma'am, biology faculty at RPS group of institutions. My dear students, we have a lot of activity in our chapter life process. Among them, one activity which we are going to discuss and perform in our lab is to show the presence of the stomata. Our activity is to prepare a temporary mount of leaf peel to show the stomata. Now, what material we required in our laboratory? The materials are leaf, any potted plant, forceps, needle and brush, glass slides and cover slips. You know it very well. Glass slide to place the sample, cover slip to cover the sample to prevent it from the contaminations, and stain, obviously the important one, saffroning. It will give the color, it will give the contrast so that we can visualize it under the microscope. Ultimately, the microscope. Blotting paper, watch glass and the water. So, by using this material, with the help of our sample, how we are going to perform this experiment? Here are the steps. First, we have to extract the peel. Definitely from the lower epidermis, as it has the maximum stomata. Staining, we have to give color to the stomata so that we can see it clearly through the microscope. Now, after staining, what we have to do? We have to transfer the sample to the slide. Then, we have to use glycerin. Why glycerin is important along with saffronin? It will keep our sample in the moisture environment. Now, after mounting, we have added the glycerin. Now, what we are going to do? We are going to observe it under the microscope. Dear students, now we are going to prepare a temporary mount of leaf peel to show the stomata. Now, we have our kit. And in this kit, we have material required. First of all, we have the forceps. Now, this forceps will help to peel off the epidermis from the leaf. We have the leaf sample. We have the dropper. Now, these two droppers, they are used for one for saffronin and another for the glycerin. Now, we have petri dish. This dish is having water. We are going to place the peel so that it will have the moisture content. Another, we have this needle. Needle is used when we will place the cover slip over the slide to remove the air. Next, this is the slide where we are going to mount the peel. Another one, this small rectangular shaped slide, we called it the cover slip. We will place it over the sample. Scissor if required. Now, this is the sample leaf. Now, the best way is to take the peel is just fold it like this and crush like this one. Now, with the help of forceps, gently remove the epidermis. When you will remove the epidermis, what you will find, you can find like this layer is, you are going to see this one, this layer is epidermis. Okay, this is the lower epidermis where we find the maximum number of the stomata. Now, in the material, you can see this is saffronin staining dye. As in the theory we have already discussed, this will give contrast to the sample. Sample means the leaf epidermis. Now, the contrast will show up the stomata. This one is the glycerin. It will keep our sample moisture. Now, one by one, we are going to perform this experiment. First of all, we have seen that this is our peel. This is the lower epidermis. This one is the lower epidermis. This one. It will have the stomata. When we will dye it with saffronin, it will show the stomata under the microscope. Now, what we will do, we will keep it into the petri dish. This one. Here we have the water. It will keep it moisturized. And then, now we are going to take one slide. This one is the slide. Now, the thing should be in mind is that this slide must be very cleaned, free from the dust.
And we should carry it from this side. Note this side. We use it keratin from these sides.
Okay. Now, what we will do? We gently take the peel and we will place it over the slide. This one we have kept over the slide very gently. Now, what we have to do now? Now we have to put some drops of saffron in. Ma'am, please take it. We have to. This is the saffron in dye. No. Now, it should be keep in mind that you should not use a lot of saffron in dye. Only 1 or 2 drops. Now keep it for 10 to 15 seconds. Yes. Now the extra dye, what we have to do? We have to remove this extra stain from this sample. It means what we are doing? We are just washing our the sample for the excess of stain. Okay. Now please. The simplest way to remove the excess stain is just pour it outside like this. Yes. Now we have only enough saffron in, no excess of the saffron in. Next, now we will keep 2 to 3 drops of the glycerin. For this sample, only 1 drop is enough. That's all. Again, we will keep it for 2 to 3 seconds. Now, here we have the cover slip. Now, what is the function of cover slip? See, with the help of this needle, I will press it gently. It will do 2 things. It will remove the air from the sample sites, and it will also remove the excess of the saffron, excess of the water, or excess of the glycerin. Now we will keep it for 5 minutes so that it can dry. Then we will see through the microscope and we will observe the stomata. So we will keep it for 2 to 3 minutes like this. Okay, we have prepared our slide and now we are going to see it through the microscope and let's examine what we observe. Did we get what we want or not? Okay, dear students, we have performed our experiment and what we get is the stomata. We have observed the stomata. The stomata have the guard cells. These are the kidney-shaped guard cells. The guard cells have the chloroplast and the nucleus. These guard cells from the inner side, they are thick. From the outer side, they are thin. Now, the space between these 2 guard cells is stomatal pore. Now, the stomatal pore and the guard cells, this is called the stomatal apparatus या aperture. Now, finally, we conclude that the epidermis have the stomata. I hope you have learned it very clearly. So, hope you have enjoyed a lot and learned a lot. In next sessions, we will meet again with more exciting experiments. Thank you, students.